So hi, my name is Ohlin. I'm chairing the Inferred Allele Review Subcommittee, IARRC, and let's see here. So we were formed originally at the 2017 meeting and then started our work in 2018 to capitalize on this combined strength of new bioinformatics tools and the information content of NGS AIR data to enable discovery and documentation of new germline gene alleles, initially in humans. And we are built on the at that time relatively recently developed tools that are now well established in the community. And we had now at the 2020 meeting, we set a number of goals that I will go through our activities on. And our core objective is, of course, to evaluate and ratify novel human alleles of immune receptors. And we have been focusing mostly on the immunoglobulin G alleles. And this is a complete list of all the novel alleles that we have affirmed over the years. And during this past period, we have affirmed 9 novel human immunoglobulin alleles, and there are more in the pipeline. And we are also, as you'll see later on, focusing a lot now on T-cell receptor alleles. We have also been focusing on trying to develop tools and guidelines for evaluation of the quality of submitted data sets, and some of that work is also done in collaboration with the software group, for instance. And we are also streamlining the procedures for submission and assessment of human inferences. So William has been working heavily on establishing processes for ENA submission together with at ENA, and also now provide updated submission guidelines at the OGRDB website. And importantly, that site also now provides a lot of supporting information, supporting new inferences and the quality of the data that is underlying new identified genes or alleles. And we can use that heavily in our internal work to build trust in new inferences. Also, AIRRC6 in Gurjari's lab in Israel has been developing a functional groups reference book that we can also use internally to support our decision making and to look not only on individual data sets but on entire population or many data sets in parallel. So this is an example of such data that represents alleles or identified alleles of a germline gene 1-2 and how differently they are expressed in repertoire. So one can really get a good picture of the data and identify spurious results if they are identified. So here is another example of 2 alleles of heavy chain germline gene 461 and new variants of those alleles. And first of all, one can realize how differently expressed they are, and that's something what one needs to understand in terms of when assessing the data. So alleles of the O2 alleles are expressed at much higher level than alleles than variants of the O1 allele. And importantly, this tool also carries a lot of additional information that can can help us to visualize the validity of the data, in this case, in terms of haplotyping information, to help us understand and have trust in the underlying data. We have also engaged, because quality is a key interest to us, and we have engaged in public discussion on different processes, in particular short read sequencing data for use in discovery of new alleles, and we want to make sure that the community as such understands the pros and cons of different technologies and the potential risks of using short read data in a very repetitive locus that is not like in this case the heavy chain locus. We have also been heavily involved in establishing a human TCR inference and approval process. That has been done within the context of the current IARC, and we have not yet formed a separate entity for this, but we have been working heavily to bring in additional expertise, in particular in the form of Jamie Heather, who is somewhere here, I guess. He's over there. And we are very much interested in getting in touch with additional members with this particular kind of research focus. So please talk to Jamie about your interest in this context. And we have done, so we brought in Jamie to initiate the knowledge transfer process to be able to maintain a high standards independently of if the work is being performed in the current IARC or if it's going to transfer to another unit. But we have already seen 10 new alleles being submitted, and extensions of 2 incomplete alleles are also, and this work is currently in process. It has not yet gone to the International Union of Immunological Societies, but that is going to be expected in the next couple of months, I would say. And there are some challenges with respect to TCR inference, in particular with the 3' end of the genes, and those are similar to what we experience with the immunoglobulin light chain genes. And we have also on our table to potentially establish IARCs for non-human species. That has not been done yet, but we have been working on these aspects together with a Germline database group, and in particular, William has published mouse IG Germline sets on OGRDB, and there has also been a paper being published in collaboration with the Germline gene database group, and macaque Germline sets are in process, as we heard recently. And William's efforts in terms of OGRDB to publish all of these the mouse Germline sets are well in place, and in particular, he published their Germline set from BALB-C that will invigorate and make analysis of air data from these from such mice much more valid. And human Germline gene sets are in progress, and something that we hope to publish soon as well, to identify genes and alleles that are truly expressed in and should be considered for in an analysis platform. And we also are very much interested in developing strategies and have efforts to work on sustainability of IARC, and we are very much looking forward to having new competence on the team. Many of us has been on this effort for several years, so staffing is a key issue here, and also to bring more competence into the TCR field, as I noted earlier. We also need to build and to continue to work on infrastructure. Currently, that's work that's being maintained by William, and of course of data to have sustainable availability of the data that we produce. But we are happy to see that the mouse reference Germline AIRR-C gene set has already been implemented in IGBlast. So it's there for you to use. And we have a long list of future plans that you can read in our report, but we will of course continue with our core activity. We will publish Germline gene sets and consolidate together with other groups the use of the temporary label allele naming, and then of course work with the TCR IARC consolidation system. These are the people, so we are individuals from many parts of the world, and I have been acting co-chair, and I'm willing to continue this work if you consider that to be the case. So, but I would certainly like to thank all of the IARC members. We have now held 99 meetings. So next time around, it's going to be a centennial. So, so I'm happy to take questions. If everything is clear, then I suggest we go to the next presentation.